To understand how consumer genomic and forensic genomic tools can tell which individuals are related to one another, it's important to refresh our information about how genetic information is inherited through generations. So what you're looking at here is a pedigree. This is a family of information. So I have coded these individuals according to their generation and their position in the pedigree. And as you might remember, squares indicate males and circles indicate females. The colors that I'm using here are just to signify the genetic individual in this, what I'm going to call grandparent generation. Um, so you see here, this grandparent generation, I've given the Roman numeral 1, and then I've numbered them 1, 2, 3, and 4 as you go across. The offspring of this grandparent generation, I have given the Roman numeral 2 and then numbered them from left to right as well. So this couple had a son, this couple had a daughter. This son and daughter then had 3 offspring, one daughter, a son, and another daughter. And I have coded this generation Roman numeral 3, 1, 2, and 3 as you read across from left to right. I've left the colors blank in the parents and the offspring generation, generation 2 and generation 3, because I want to work through some examples so we can remember how chromosomes divide and recombine in order to form gametes, which eventually form offspring in sexually reproducing organisms. Let's start with the first pair of grandparents, the ones that I color coded pink and yellow that had this son, which we haven't color coded yet. Let's think about one chromosome of information that is in the gametes of those grandparents that formed their son. So I'm going to color code that as one pink chromosome from that grandfather and one yellow chromosome from that grandmother. So if this is one chromosome of information, present in the gametes of those diploid individuals, those gametes fuse, and now we have the homologous chromosomes of information that formed their son. So now I am going to color code their son as having one pink chromosome and one yellow chromosome. And we do the same thing for the other set of grandparents. This is one chromosome in the gamete of that grandfather. This is one chromosome in the gamete of that grandmother. Those come together. These represent one homologous pair of chromosomes in the daughter, which we can now color code as red and blue. And now the question is, now that we have our parent generation here, what are the possibilities of chromosomal combinations in their offspring? So let's start thinking about some potential offspring. Let's create their first daughter, Roman numeral 3, numeral 1. So what you're seeing here, I've divided by this black cord, are the homologous chromosomes of the dad and the homologous chromosomes of the mom. So the first thing that needs to happen when these parents are forming gametes is that the cell will duplicate the information on both of those chromosomes. That's going to give us that characteristic X shape of the chromosome that is associated with a mitotic cell in the middle of cell division. And then, of course, we're forming gametes, so we're going to have recombination. Somewhere along the chromosomes, these homologous pairs are going to line up. Well, let's do it in another random spot. Let's say that it happens down here for the mom. And those chromosomes are going to exchange information. So recombination between homologous chromosomes occurred in the gametes of the father and the gametes of the mother. And now we are going to form the haploid gametes that will eventually come together to form their first offspring. So I'm just going to separate these pieces of clay. That's indicating that we are forming those 4 haploid cells. And here comes another random bit. That is that any one of these gametes can come together. So I'm just going to randomly choose 2. I'm going to choose these. All of the rest get discarded. Nothing happens with them. These gametes form together. They're no longer in a separate organism. They have just formed their first daughter. So now I can color code her just based on randomly what I chose. She has one chromosome that's very, very much like her grandfather, and one chromosome that is very much like her, who was that maternal grandmother. and paternal, a little bit of paternal grandfather. I'm going to repeat that same process to form the other siblings. So just again, these are the gametes forming in the dad, the gametes forming in the mom. The first step would be duplicating those chromosomes while these gametes are forming to form that X-shaped mitotic chromosome. We'll have a recombination event somewhere randomly along the chromosomes. There will be a recombination, and then those chromosomes will be randomly reassorted into the offspring. I'm going to stop the video here while I do these, but I'm going to show you the progeny that I randomly create in just 1 second. So here are my results of random recombination events and random segregation of chromosomes that formed these 3 offspring. A couple things to note here that these are all siblings, but they don't necessarily share 50% genetic identity. That number that you hear about siblings, full siblings, that is, siblings who share both a biological mother and a biological father, being 50% identical is really just an average across all of the chromosomes and across all siblings. But you can very much have one sibling who is more genetically related on a base by base case than to another one of their siblings. Another thing that you might notice is that not all of the grandparents are represented in the offspring. There are some opportunities here to randomly lose segments of DNA from the grandparents just based on which chromosomes recombine and which chromosomes make it into the gametes. And now let's remind ourselves of what these different colors of clay represent in the genetic information. And what these colors represent are genetic differences among these individuals. So here I'm mostly referring to SNPs. That's the conversation that we've been having this week. And remember, there are about 300 million SNPs in the 3 billion base pair human genome. So obviously not every single site along this pink chromosome is genetically unique to every single site across this mix of blue and red chromosome, but there will definitely be individual locations where you see one allele on the pink chromosome and a different allele on the red chromosome. So let's draw an example of that. Let's say that at this particular locus, I see a T at my locus of interest. But if I look on the homologous chromosome that descended from a different grandparent, remember this pink chromosome came from this grandfather I1, and this red chromosome came from grandfather I3, I might see that those individuals had different alleles at this locus. Let's say there was a G here. So if I color code that across all of the siblings, I see that at least at this locus, every single sibling happens to be heterozygous for that allele. I also want to point out that when we're comparing relatives' DNA information using SNPs, we not only care about the number of SNPs they have in common, like, oh, do they happen to be heterozygous or homozygous for this SNP? We care about the length of DNA fragments where they have shared SNPs. And what we're looking at right now as we compare these different individuals is that they all have very long shared bits of genetic information. And to us as genomicists, that means that they're closely related. And it means they're closely related because it means that there haven't been many recombination events to mix up different chunks of chromosomes. To illustrate that, let's add in yet another relative. Let's say that these grandparents in this generation had 2 daughters, and you're seeing here that these 2 daughters look to be genetically identical, but it's important to point out here that's only because I gave each grandparent a single color. But you can imagine these grandparents are not the first 4 people on Earth. They have lots of shared sequences and lots of different regions of their chromosomes that they inherited from generations before them. But just for the simplicity of this exercise, which is just to show you how blocks of genetic information get inherited together, they're going to look similar. And similarly, I have here the male that this other daughter of these grandparents is going to produce a child with has color-coded unique colors that he inherited from his parents. So we can recreate our exercise to see what is going to happen as their gametes form. And oh, I lost my chromosomes. I'm going to stop the video, but I think you get it now. We're going to duplicate the information, recombination, form the gametes, and see what does their daughter look like. So here is our result after a round of recombination and random assortment of choosing a chromosome that made up the gamete that eventually formed this daughter. We have a pattern that looks like this. And now I can tell that this individual Roman numeral 3, 4, is a cousin to this set of siblings over here. And I can tell that because I can see it in the pedigree in front of me, but also as a genomicist, I can tell. Because one thing I see is that they have an entire parent's worth of chromosomal content that is unique. And that's because while their mothers are related, their fathers are unrelated. Now, that tells me that they're unrelated, but how do I know that they're closely related? And again, I know that they're closely related because I see large blocks of chromosomal similarity, specifically along this red and blue chromosome that they each inherited from their mother. Now, these large blocks of chromosomal information, these contain SNPs with some regularity across the length of that chromosome. We call these large blocks haplotypes. Haplotypes tell you the genetic information or genotype, that is what SNP is present, but it's not only at a single locus, it's at a stretch of the chromosome. So for example, for these individuals, we might say that they have this stretch of haplotype here in the red region of the chromosome. That is, all of these individuals inherited the same SNPs along that length of the chromosome. Here are my actual 23andMe results. So 23andMe used about 1 million SNPs from my DNA, and it found other users in its database that may or may not be my relatives. The only one I actually know is this one, and 23andMe makes a guess about how related this person is to me, and it makes that guess based on the percent DNA shared, which is just overall, how many SNPs do we have in common? And then importantly, how many segments we shared. Again, by the end of this video, I really hope that you're understanding the importance of those long segments of co-inherited SNPs, which indicate a familial relationship between people. So based on that, 23andMe looked across all of those segments in DNA and guessed that this is my first cousin once removed, which is correct. That's my mom's cousin. The rest of these I don't know, but you can see that there is some correlation between the amount of DNA shared in the segments and how closely related 23andMe thinks that individual is to me. So overall, that's how we're doing it. We're not only looking at genetic similarity at a base by base position, but we're really interested in the long segments of co-inherited SNPs that indicate a close genetic relationship.